Hello everyone, welcome to the last session of the day. Remember, the best is saved till last, so now's the time to get off your phones, stop your email, and the best talks are about to hit you. So let me go to the right slide. I won't talk everyone through now. It'll be a great pleasure to introduce people as they come up. We will be thinking in this session about monitoring evidence and innovation surrounding marine artificial structures and decommissioning. I'm going to introduce myself, Murray Roberts from the University of Edinburgh, and I'm actually going to be giving the first talk today on the North Sea, and actually picking up on some of the angles that we've been looking at. And I want to talk slightly on a different topic, mention a word that I don't think we've heard yet. We may have, restoration, and I want to talk about deep sea corals that we have heard about. And one thing I'm feeling up here today is incredibly old, because Joe put up some papers that go back to the early 2000s, and I wrote those, so I'm feeling just like ancient. So don't worry, Dick, and I'm not going to ask for audience participation, but I'm one of the oldest people in the room, and this is so depressing. I want to acknowledge my co-authors, Simone, who's in the room, Rob Ogden, who's a conservation geneticist at the University of Edinburgh, and Leanne Henry. And we're going to be talking about a role for North Sea rigs in cold water coral restoration. And why are we talking about that? Okay. Cold water corals are just wonderful, and I could spend 2 hours talking to you about that. But don't worry, I'm not going to. But if you haven't seen deep sea coral videos, Google them, have a look at them. They are truly kind of the cities of the deep sea. They're vastly important for sustaining diversity. Fish spawn there, lots of things happen there. But they are nothing like they used to be. In many areas of the world, they've been trawled out, they're gone. Our baseline has shifted. So if we look at areas and we think they're perfectly functional, we've forgotten that a lot of the structural habitats that used to be there aren't there. A key area for us in the project we're undertaking at the moment is west of Shetland, where Wyville Thompson, who led the Challenger expedition, referred to stony copses of coral covering many miles. I went dredging there myself in the late 1990s. We only dredged up rubble and a tiny, tiny fragment of live coral. That was the late 90s. There's been another near 30 years of trawling west of Shetland. Now, on top of that, the world is changing. We had a brief discussion of climate change, but I think it's more than the elephant in the room. It's our reality. The habitat suitability for deep sea corals is changing enormously rapidly. I still use this older term, Lophelia petusa. There's a bit of a debate in the deep sea coral world about the term and whether or not we should have changed the species name or not. So you might hear me kind of flip between Desmophyllum and Lophelia. It doesn't really matter in the context of what I'm going to say. The key thing is the habitat suitability is declining very, very rapidly. So if we restore areas, we've better understood how suitable they're going to be moving into a future ocean. So this allows me now to introduce the project I want to briefly summarize and our role in it in the context of an interesting opportunity that arises as the North Sea is decommissioned, and that we think about whether or not we can give nature that helping hand and restore areas of the deep ocean. So the Redress Consortium is a consortium of primarily academic organizations and small enterprises across Europe. It's a European Union funded project, and you can see we're working in a whole variety of places to try out deep sea ecosystem restoration, to see if we can build the partnerships to make it a reality, work through the policy constraints there might be, develop aspects of best practice both for that process and for the ongoing monitoring of the success of the restoration activities. And I'd like to focus in on this area, west of Shetland, and I'll be bouncing this between east of Shetland and west of Shetland throughout most of the talk. And to say, as we've seen these wonderful images of deep sea corals, well, they're currently a waste management problem. Now, that's bizarre, right? That we've got this situation of 50 plus years of deep sea coral growth, and goodness, those corals are huge now. And the best idea as we decommission is, I think some places compost them, other people throw them in the skip, but they're actually a problem and a hazard for the people who are having to deal with these when they come onto shore. We've been looking at this for a few years, on and off, and you can see here a real a reef, artificial reef. That's one of the legs of one of the platforms. I'm not supposed to tell you which one, so I won't. But it's absolutely smothered, just as we've seen from Joe's talk. We've been comparing with natural reefs. We've done a lot of work of that type. The fish communities, the invertebrate communities, certainly the coral cover is more impressive on the rigs than it is on the natural reefs. It's an interesting thing to see, but what does that matter? What can we do? How can we take profit from this situation? So just to give you a few moving images and to give you a sense of just how much coral is there. Again, we've already seen some of this, but just look at the way this grows. We've actually been simulating the growth of the coral in computer models. We can generate this kind of growth form now in silico. So we're getting quite good at understanding why the corals grow where they do. And yes, it's not facing into the current. They don't like a hairdryer feeding them. They like to be kind of on the back side of things, where there's a bit more turbulent flow. The polyps can really feed beautifully in those areas. And over the years, we have been also running a whole series of other experiments, both in the computer in terms of simulations and now looking at the genetics of deep-sea corals. Give credit here to Leanne Henry and the team behind a first phase insight project that asked the question, well, these corals live on the platforms. When they spawn, and we figured out by looking at their reproductive histology 20 years ago that they were reproductively active, well, where would those baby corals go? Where would the larvae end up settling? This work is carried on in a number of places in the North Sea and other sites around the world. And you can see that the corals do spread quite some distances, including, we would say, feeding into marine protected areas designated offshore Scandinavia. So one of the really interesting angles now is, fine, we know this from computer simulation and from great work that's looked at the larval biology of these corals, so we know a lot about the competency, how long those larvae spread in the water column. Can we back this up with genetics? What do we know about the genetics of these corals? So here you can see people collecting coral kits. We've actually sent out coral kits just in the last few days to go out to the North Sea platforms. And anyone that wants to do this, please contact myself. Leanne, Simone is in the audience. We will send you coral kits. And all you need to do is pop a tiny couple of fragments of this coral into an Eppendorf in ethanol, send it back to Edinburgh, and we'll give you a readout of where your corals fit in the genetic mix of the platforms and the wider area. And why does that matter? Well, the first thing to say is we're not going to go restore a place, moving corals from one place to another, unless we know it's a genetically sensible thing to do. We certainly don't want to cause problems. So I want to talk you very quickly through what we know of the genetics of deep-sea corals, this particular species, Lophelia or Desmophyllum pertusum. And without diving into all the gory details, there's an isolated population in the Mediterranean. They're somewhat genetically distinct. It's isolation over space. There's a boreal population that we have evidence for. It spans quite a wide area, but that overlaps closely with this. This is the Lusitanian distribution, wider one, and then this boreal distribution up here. Okay, there's a lot of mixing. Is the take home. From these corals, and the North Sea populations sit right in that mixing zone. Very interestingly, when we had enough corals to do a preliminary, very, very first preliminary analysis, which was done through the context of these European projects a few years back, we found that some of these platform corals actually came from several places. You can see the names of the platforms here with some slightly eccentric French spelling, but you can see there's a bit of a mix, right? There's thistle seen here and there's thistle seen over here. So they're actually like settlement plates, right? They are basically settlement plates. And over those years, corals from a variety of places have settled on those platforms, and they are spawning, they are active, and they are spreading like wildfire. So why is this important, and what can we do about it? We go, and there's just a little zoom into those sites. We've also, just in the last round of sampling, been able to get by happenstance and by catch. I love Youp's machine for sampling the invertebrates that live around the platforms. When I first sampled deep-sea corals from the platforms in 1999-2000, it was just the coral I had in my hands. There was barely any other associated fauna. And as you saw, the corals were teeny tiny. Now they're vast, the size of this screen, 1 or 2 colonies. And they are full of associated species that we weren't seeing in those few samples. So wouldn't it be exciting to get out and properly sample and inventory the diversity of these other classic associates that you see on natural reefs? So I want to give you an example of how this is going. So here's a collection of live corals from the rigs going on, and another little video, which I think is rather an interesting picture of this very sophisticated scoop that we had built. And you can see the scoop about to do its job. Watch out, Mr. Crab. Your home is about to be rudely removed. A little shuggle, and I think it looks like we might, Simone, have got the crab as well. But I didn't see that in your collections. It fell into a Zarges box. This is the low-tech, basic level entry way of getting the samples. But from this, we have our live corals, which we've transported from the ship offshore back to a marine station on the east coast of Scotland near us here in Edinburgh. It's the St Abbs Marine Station, where the corals are in the tanks. And here you can see polyps expanding, and the whole thing now is keeping these precious corals alive for long enough to, for the first time, actively restore a deep-sea hard coral system west of Shetland we're going to go to, which has been largely damaged by trawling. So taking you into the logic of what we're doing next, restoration ecology is talked about in a variety of ways, okay? Passive restoration would be your sort of entry point where you simply close an area, let it passively restore and come back. Now, the marine protected areas around Scotland, including the Spongebelt MPR, are still trawled, actually. So that's going to take some time. Maybe there'll be trawl closures brought in over time. But I would argue that giving nature a helping hand, as we do in terrestrial environments, is a good thing. And why not take profit of that 50 years of coral growth to restore areas? So we're in the process of now planning the details on this to restore in this southern area of the Spongebelt MPA in waters suitable for deep-sea corals, using these 3D printed reefs where we'll put live corals onto these and put them out actually in the footprint of the Foinaven field, which is closed to trawling within the 500-metre trawl exclusion zone, working with BP later in the summer. Now, one of the key things that I want to talk about, and I took the slides out in the interest of time, but this area and the areas in the North Sea, when we look at the suitability of the habitat, remain suitable for deep-sea coral growth many, many years into the future. So the deeper areas of the Atlantic are going to become corrosive to deep-sea coral skeletons by the end of this century, and many of them are going to see a tremendous decline in the food flux. The productivity from the surface won't be nearly sufficient to feed the corals that grow at depths, at around 800 meters water depth. The shallower areas, like the areas west of Shetland, like the areas actually in the North Sea, are going to remain highly productive with strong food flux. So part of the research program that we're undertaking in Redress is following that through. So we identify the climate refugia. They need to be resilient to ocean acidification. They need to be with sufficient food flux and not becoming unsuitable to deep-sea corals in the future. And it's that food flux that's really, really critical. A lot of restoration projects are going out there and not necessarily considering that whole suite of parameters. And just for those that want the details, that work's being done with Yuri Artioli at Plymouth Marine Laboratory. So to kind of wrap things up from my side, I think we have some really interesting opportunities here. But the scale of the challenge of any ecosystem restoration is huge. In the deep sea, it's ridiculously vast. Academics can kickstart and catalyse. Then we have to pass to industry and government for future. And I think in this project, we have a chance to really show how that could work. If in the future there are funds, novel funds that are created, novel ways by which we can monitor and sustain, for instance, MPA monitoring, maybe there's some clever thinking that could be done in those areas. Right now, we're restoring within existing industry footprint. That could be offshore marine renewables into the future, of course. But building these partnerships is key, and I think it ties really well into a number of important policy imperatives. We are in a decade of ecosystem restoration as well as a UN Decade of Ocean Science for Sustainable Development. I will leave it there and thank everyone who's involved, and there's a roll call of the partners involved in the project. Thank you very much.